It was previously proposed by Ino et al. Study that the Malagasy River limits gene flow due to the lack of shared haplotypes between the north and south banks. However, the authors of this paper aim to evaluate the Malagasy River and prove that it is not an absolute barrier to chimpanzee movement, and as a result, chimpanzees either side of the river should be considered a single population. To investigate this, 11 highly qualified scientists collected and recorded data in regards to movement, fecal samples, vocalization, and nest locations on several chimpanzees in the Greater Mahale ecosystem and the Greater Gome ecosystem. 16 fecal samples were extracted from the north and south of the river and subjected to mitochondrial DNA analysis to confirm the origin of their species. The D-loop region of the mitochondrial DNA genome was amplified, and as a result, 13 samples yielded 3 different mitochondrial DNA haplotypes, all of which had been previously identified to cluster with eastern chimpanzees' sequences. This discovery has been visually displayed in Figure 3, showing the relation of the 3 species to previously identified chimpanzees within Tanzania. All genotype fecal samples were subjected to Simian immunodeficiency virus detection and also screened for HIV. This procedure involved analysing the presence of antibodies using a highly sensitive and specific Western blot approach. As seen in Figure 4, 2 chimpanzees exhibit clear evidence of Simian immunodeficiency and HIV infection, MR1349 and MR1343. A previous study conducted by Rudisell on SIV suggests that some mitochondrial DNA haplotypes from the south and the north correlate, but it was unknown whether this was due to recent gene flow or distant common ancestry. The results shown from this particular paper indicate that recent gene flow is in place, as the 2 infected subjects were found from opposite sides of the river, ultimately suggesting that the locations are epidemiologically linked. Another strong piece of evidence suggesting that the Malagasy River is not an absolute barrier was the discovery of a natural shallow ford, which was able to be crossed on foot by investigators. Whilst under examination, 2 fecal samples, both obtaining the characteristic size, smell, and shape of chimpanzee feces, were identified within the riverbed itself. This particular finding strongly suggests that chimpanzees are in fact able to cross the suspected barrier. The fecal samples gathered within the riverbed and on the northern and southern banks do not provide conclusive evidence for connectivity between the 2 regions. However, the discovered ford does supply an explanation for the shared mitochondrial DNA haplotypes. Further field studies need to continue to provide information on the gene flow of chimpanzees within Tanzania.